That computer giving you problems still? Or is it? Oh, no. Okay, let's talk about a couple different ways we can approach some of the cell organelles. Um, the one thing just to think about if we're uh, talking about lysosomes, if you take a cube and smooth the cube, sorry, let's see here. You can pretty easily delete half of that and then extrude out a section. So if you're trying to do a section of a lysosome, you can do that. Now, one thing, and this is a really, really minor um, nuance here when you're turning a cube into a sphere, is that it's not exactly um, spherical. Um, it's pretty close, but you can see that the poles are sort of pinched in a little bit. So if you want to rectify that, um, one of the things you could consider doing is just using a deformer like your lattice with an extra subdivision on each pole. And if you just scale that out ever so slightly in each axis, you can round out that object. If you, at this point, then delete your history, you'll have something that's just a little more spherical. I don't so much worry about the backside. You could actually pull the backside of it down a little bit too, just to round the back of it out, but it's really more the getting the edge to look spherical. And then obviously if we want the edge to look cut, we could go in with our edge loop tool and just add something close here. To give it a little bit more of a cut look to it. It just, like I said, it's a minor subtlety, but just taking an extra second to just pull it out and round that off a little bit. Just looks aesthetically a little nicer. Okay, so rough ER, starting with our friendly cube. Now, what was the word cisternae, which is the pancake stacking type thing. Rough ER can be pretty similar. I showed you sort of how to do that with the Golgi, but the difference with the rough ER is that things tend to be connected. So the parts are connected to each other. So instead of being able to round out a cube, we need to think about sort of the scale here. So let's start by making this 12 and maybe a height of 8. And We want enough subdivisions in this to be able to sort of make connections between the neighboring components, but not so many that it looks like a rounded off rectangle. So let's try to make this kind of square. Um, but maybe what I'll do with this is make some modifications just to kind of round things off a little bit. So that if I look at this, it looks, I guess, a little bit more like a inflatable something or other than it does look like a perfect rectangle. Um, I'm thinking about this in terms of working with it flat, not thinking about the curvature of it at all, because it'll make it a lot easier for us um, to modify the faces and connect the faces. And then we'll use our deformers at the end to bend it into the shape that will fit around the cell or the nucleus. So um, I'll show you how to get started and then kind of leave it up to you to work out the details. But we could take a um, couple things here. Maybe we want to scale parts down just a little bit. We could always make more shapes um, here too. So let's make another version of or another piece of this as well. And I'm pulling these kind of far apart for now. We can make them a little bit tighter once we uh, bridge the gaps. But um, what I want to think about is, like, if I were cutting this in half, where would I cut this? And if we look at just where things fall, there's a nice dividing line right at the equator. So we can work, if we want to show a connection where we're cutting through, we might want to pick a face that's on the equator, like maybe take something here. And I'm just going to extrude that out a little bit. And then we'll find the neighboring face over here. Looks like probably this one. And I'll extrude that out a little bit as well. Now, in order to connect these, one of the key things is that the objects have to be the same object. So we need to take the two shapes and say combine. Okay. And that combines it into a single object. Then we can go and delete our faces to create a hole. And I know you can use the bridge tool to do this. Sometimes it works fine. I can double click here and say bridge. And in this case, it worked all right. So, we'll take advantage of that. If for some reason bridge doesn't work, the other way that you could go about connecting this would be to select vertices and use the merge vertex tool. And I just want to double check the threshold on this and make sure that it's large enough that if we merge those two vertices, it will occupy this or expand over the space and distance that the vertices are placed apart. I personally like using this method over using the bridge only because then when I'm done, I know that this is connected correctly. So we've created a bridge between the two here. We could maybe do a lower one just to make this more interesting. Maybe I'll take one down here. And find kind of a neighboring in here. And again, I left a little bit of space in between these just to make it easier for me to isolate. And see what I'm doing. If you get to a spot where it's hard to select, too, one of the techniques you could use would be to isolate select like that. And then that makes it a lot easier to figure out what's going on here. So vertex, the vertex, and you're less likely to make an error if you don't have so much stuff to worry about behind you. So we got those two merged together. Turn off view selected. And because we combine these surfaces now, when we smooth that, you can see it gives us kind of a nice connection. If we take this shape now, and I mentioned the other day when we were talking about this, thinking about the overall flat surface of everything first, because it helps you to keep track of everything. I can tighten these up now a little bit by selecting my vertices here, moving this a little closer together. And I can even snap align this row if I want to make it easier to select. Once I've got that in place, I can go into my side view. And this is a case where I'd probably, if I want to keep the whole one of these two, I'd probably keep that and duplicate it. So I can always go back to it if I need to, but this one will take and delete our faces here and probably just take these out too so that everything rounds off nicely. I can preview it and see what it looks like. You guys can kind of see where we're going here. So taking the time to just build that out with some different size structures can pretty quickly give you the base for the rough endoplasmic reticulum. And then once you're ready to bend that, you can use your bend tool. And you can see how that starts to take on an RER type shape, right? So if we then wanted to add some ribosomes to this, let's zero out the bend for a minute. I'm going to duplicate that, hide these, and we're going to talk about something. If I'm trying to remember, did we talk about, has anybody talked at all about mesh networks? Did we talk about that in any classes? Okay, all right, that's fine. Basically, the mesh network is a way for you to create instance objects. So you're taking a replica of a single object and you can disperse that in space in a variety of different ways mathematically, or we can actually use the tool to disperse things over the surface of an object. So the way the MASH works, let me just get a little save here. Is that you create an object and we're just going to create our cube and smooth that. We'll scale this a little bit smaller for now. I'm going to select my object, and the key when you're creating mesh networks is you want your object, your source object, to be on the zero axis with zero transforms and zero rotation, that sort of thing, because the placement of the object in space is ultimately going to be dictated by the mesh node. So you want everything to be zeroed out first. If you have translation values or rotation values, those will be applied on top of the mesh network, and you may not get the results that you want. So the way we get to the mesh network is to go either under animation or the FX menu. And there's this menu that will come up here that says mesh. And we're going to say create mesh network. Okay. And what generally happens is that you'll get a replica of the object that you selected for your mesh network. And it will just kind of go out here into space. So what can we do with this? This is where things get really fun. And after I show this to you, and after we work with MASH for a little while too, you're going to start seeing this pop up everywhere. If you look at commercials and things like that, you'll start seeing the stuff, and you'll be like, "Oh, I know how they did that now." And there's an equivalent tool in Cinema 4D. I'm drawing a blank on the name at the moment, but basically it operates the same way that the MASH network does. So you create instance replicas of things, and you can do cool stuff with them. So the mesh node itself pops up in your outliner here. You can rename it if you want, but the distribution is kind of the key here as far as looking at what you can do just at a base level. First of all, you can control the number of points, so we can make multiple objects. You can control how it's distributed. So when this says spherical, that means it's basically making an imaginary sphere and then distributing the objects over the surface of that. Okay. We'll talk about mesh in a second. That's the one we're going to use to put these onto our object. but you can also create them in a grid and control the number of. You can really do a lot of cool and powerful stuff with this and have a lot of fun with it. We're just going to scratch the surface today with this stuff, but we'll keep talking about it and look at different ways that we can use the mesh network. So what I'm going to do here is I want to distribute the mesh network over the surface of the RER. Now, there are some limitations. You can do this randomly, but sometimes as artists, we want to have control over where we're placing things. And so we can utilize the face center as a target for our mesh network. But if I look at this and I think about like how many ribosomes I could put over the surface of this, if I'm targeting the face center, there's not a lot. So this might be a point where we would want to smooth our model a little bit further so that we can have more choices for a target. So I'm going to go back to my modeling menu for a moment and smooth this 2 more times. Now, I know we've talked about wanting to minimize the amount of geometry that's on your objects. So you may want to experiment here. You don't want this to be too dense, but we also want to get enough faces here so that we have something where we can sort of randomly select faces so that we have some control and don't have a direct or exact pattern. And I know this might seem a little tedious, but taking a few minutes to do this by hand, or sometimes you can find scripts and things too, where you can just have a random face selection by plugging in a little script from an online source to speed this process up. If you only have to do it once or twice, it's not the end of the world. So I've created a couple faces and selected those faces. I'm going to do something here called create a quick select set. And in doing that, I'll just call this ribo target. So you'd want to take the time to go around your ribosome. And because we're trying to limit the amount of polygons too, maybe you just want to go around the outside of it and kind of ignore the inside, or maybe you just want to put a few on the sort of on the inside there as a target. But I can always now recall these faces by selecting this in the outliner and saying select set members, and that will recall those faces. So now we want to take our mesh network and we're going to want to set the distribution to mesh, okay? And this will immediately bark at you and say there's no mesh connected. So what we need to do is middle mouse, drag on the object. And when we do that, you can see that that locked the spheres to the surface of our object. And we might, if we increase our points enough, we might be able to get pretty good coverage on our own just using the scatter. But I don't particularly like the way that looks because it's just not, it's not separated out enough. They kind of block together. So what you can do to control this is we can choose the method to be face centered, in which case it's going to just cover one of these right now. But you'll notice over here there's an input node for selection set. If I take that and take my selection set that we just created and send that over here, now I'm actually putting the ribosomes onto the faces that I selected. And that's giving us a little bit more controlled distribution. And you'll notice too that these look like they're sort of halfway inserted into the object. But one of the cool things about this is there's a node called push along normal, which starts at 0. But if I increase that, you'll see that I can actually push these out relative to the surface and get them to sit right on the surface of the object. Okay. So then, let's see. With those sets selected, I think I can still bend this, but let's do that. Now we've got the RER is still, or the ribosomes are still attached to the surface as we bend it. And then we could do our final extrusion on this object. Oh, that's interesting. Oh, that was an interesting side bit. Probably something we don't want. Good to think about that. So when I extruded, it actually also flipped the selected set and assigned a set of um, ribosomes to the inside of the ER, which this you might not want. So there's ways we could still work around this, but that was just curious. So if once we have things the way we want them, we've got the ribosomes on the outside. The repro mash node here, or repro mesh node, is basically the polygonal equivalent that's created using the MASH network. And if I take that and duplicate it, that's going to break all of my connections to the MASH network. So it's basically locking it in now, or uh, committing to it. And if we just see, we can select. If I just have faces on and double click, I can actually delete anything that I don't want. So this would be a way to deal with something that's inadvertently comes up as a result of extruding the surface. As long as you've got objects that are independent, you can kind of go through and oh, and these are doubled up too because the number of nodes I had exceeded the number of faces in my selection set. So that's why we're getting two at some of these nodes. Anyway, you get the idea there. So if you do get in a jam with the mesh network, just know that you can reproduce the mesh and you can edit it as you might need to. So now we've got, this would be our ribosomes. This is our rough ER. And it's, you know, this isn't the lowest poly count piece by any stretch of the imagination, but it's not so heavy or high that it's going to break a system. And I should say, once you commit to things like this too, you can, that's the time, once you're happy with your model, is the time to delete your history, because none of that will carry through anyway into your um, Sketchfab piece. So let's call this group that, call that RER. And let's talk about smooth ER. There's been a couple different ways that I've tried to model smooth ER. One of those was to start with the cube and make a shape that's kind of like almost like a jack, like this. And then from that, you can duplicate. And you could use your tools to connect these together. It seems, again, some of this stuff seems a little tedious, but if you keep it really simple, you got to take these shapes, combine them, and then we can merge our vertices. The idea is once you get a block of these together, um, you can then, uh, because the shapes are pretty simple, you can sort of move things around a little bit, make them look a little more organic. Let's maybe add let's add a couple more onto here. Um, Notice I was picking through on a couple of those and had to tumble around to catch that. But So you start getting some shapes together like this, and then once you have one shape, you could always duplicate that shape too. So you sort of build larger shapes or larger blocks, and then start to, if you're trying to add some uh, organicness to it, you can kind of offset things, move things around. Um, but the idea here is that you start to get something that's got a little bit more of a tubular structure. When you start connecting things together a little differently, it takes on more of an organic shape. Um, I'm not, I was satisfied with that for a while, but I kept scratching my head thinking there's got to be a little better way to do this. And so a different way that I found to make your smoothie was actually start with a torus. And if you reduce the subdivisions, to something that's fairly square, um, if your subdivisions around the height are 4, that makes a cube. And if you rotate or twist the radius, it sort of it makes a shape like this. Just to keep track of things, we'll go ahead and rotate this on the axis. So we've basically got a ring of cubes now. And if I duplicate that and move that over a little bit, you can see where we can start to build a little bit of an interconnection. You have to, again, combine those so that we can merge them together. Try the bridge tool again. And what you see is we start to, you start to get something that pretty quickly, if you scale these or move these things around a little bit, you get some interesting structures that are also interconnected. We can take that shape, and if we get rid of our upper faces here, then when you smooth that, it starts to look like a bunch of macaroni noodles together. But in looking at different representations of smoothie I think that's not too far off. So you could maybe want to offset these or move these a little bit. Scale them up or down a little just to give some variation. And pretty quickly you can start building the structure out. Once you have one and you've got some that are interconnected, you could always duplicate that. And maybe we take these and combine them here and try our bridge tool again, see if we have, and that's not too bad. So I just got rid of the faces and bridged that. But if we, start to look at it, the structure, starting to look fairly organic, and it's a good beginning of what I think the smoothie R looks like. So um, maybe not as, if we were to take some faces off of these here. Oops. It's kind of a little different approach, a little different structure to it here. I kind of like the way this is shaping up better, and it seems, from what you're studying in histology, you'll probably find this to be a little closer to the structure of your SER. So two different approaches on that. Let's see what else. Yes.
Here, just on the like more macaroni shape,
Mhm.
would you just extrude it then?
Oh yeah, I'm sorry. Yeah. So once you get your shape the way you want it, then yeah, you would do the same thing here. You could take different faces and extrude those. And you'd have to play around with whether it works better to extrude it out or extrude it in. But either way, you should end up with something that gives you a pretty good representation here. And you could use the same technique of adding edge loops at the end of it to kind of flatten those edges out. I mean, just to make that point, we can do one here and so you can see how that's got a little bit more of a cut to it than this, which is a little more rounded. But yeah, the whole concept of this is just thinking about like what is the shape that you're trying to create, creating that in one single layer to the best of your ability. Think about it as the whole first, and then save that piece, cut it in half to give you your cut section if you're trying. Because that way, if you want whole Golgi in the cell somewhere else, you can put them in the cell as a whole, but then you can also have like a cut section if you want to zoom into that or put it in your cut section of your cell. Same thing with these. It's really just the process of like, how can you take something and simplify it to the lowest common denominator that you possibly can and still get the result that you're looking for. Um, let's maybe do one more. Let's talk about mitochondria for a minute. Now, mitochondria come in all sorts of shapes and sizes. So, um, looking at the cell that you've got and understanding what the mitochondria typically look like for that cell is going to be your first step. But for the purpose of the demo, let's go with the sort of generic kidney bean looking mitochondria. Let's see, I'll start making this a little bigger here so we've got some room to work. So if I want this to be taller, okay, if I smooth this, this is just kind of giving me a preview of the shape, and That's not too far off. It's a little more pill-like than kidney bean, but maybe if I add more subdivisions, it's going to be more pill-like. So maybe I want to, um, maybe I just want to scale these ends a little bit. That's a little better. Kind of round it off a little bit. And again, I'm thinking about this in terms of the orthographic shape that I want to start with, because I know I can bend it later, right? So I'm going to build the mitochondria as if it were straight up and down, and then we can manipulate it after the fact. So to if I'm reasonably committed to this, then I could actually use the smooth tool, and maybe since I want to cut this in half, um, I could grab half of this and cut it. Oh, and I should, if I were smart about this, I'd probably want to duplicate that first so that we've got our hole, right? And just get that out of the way, and then we can do. So now we'll make our cut. And this is just going to help me for a reference too, as far as how we want to make the inner structure. So remember, mitochondria have two cell walls, right? So we've got the outer cell wall, and then we've got the inner part that make up the cristae of the mitochondria. So we want to think about creating another cube for the center, and we probably want this to be a little shorter here, but maybe 3 and 3 here. That looks like it's a pretty good fit on the inside. And we're going to want to make a bunch of interconnected discs with this, so we need some subdivisions here, or yeah, maybe smoothing this would work out okay too. So we could say smooth this structure. I don't need threes too many. Two might be a little better. So let's go with that, but I want to make it a little bit thicker and also just a little bit closer to the circumference. So if we take this now and duplicate it, and maybe we move it up and scale this down a little bit. I'm trying to think about the 3D structure of what these cristae might actually look like in the cell instead of what we're used to seeing in the cut section. But we can take these and combine them. And again, think about like, well, they have these structures have very, that's going to get in our way now. We'll just hide that. These structures do have kind of an organic quality to them that we want to uh, try to maintain. If we're going to cut these, though, we might want to do this somewhere where it's going to be near the equator. So let's take those, delete the faces, and see if we can't get an edge loop that'll cooperate. For our bridging, that looks pretty good. I think you can start to see where I'm going with this. It's, you're just kind of making some really interesting uh, sort of sci-fi structures here. turn that back on for a minute, just make sure we're in the same space. I could actually take this and duplicate the whole thing, so I don't have to recreate that one bridge. And maybe we'll scale, reverse the direction of that to give us just little more variation. And let's, since I did the, reverse, in the axis there, that's one time where I would actually say it's a good time to freeze the transformations here on this one. And let's, combine these two. Combine, and let's do another. Let's take.
Yeah.
Did I put a hole? I guess I put another hole in that too. That's interesting. Okay. Just caught that. That can be fixed. Interesting. All right. So every now and then things get a little wonky, even for me. And it looks like for some reason here we're having some things that aren't going to cooperate. I see that I've got this hole here too that might be giving us a little bit of a problem. So we can fill that hole. And I don't know where that came from exactly, but there's another one up here. All right, that should fix that. And just for the sake of the demo, let's just try to simplify this by catching neighboring faces there. Must not like it because I flipped it. Let's see what happens here. This may be a place where the bridge blows up. Yep. Okay. What was our error message there? Oh, dang, we're not combined, so I must have backed up too far. So that should combine. And we got those. Let's see if we can bridge that. And now it doesn't like it. Okay. So in a case like this, we could say extrude our edges. Yeah, there's some weird stuff going on in here a little bit. And it's good to know the merge um, technique too. Oops. Because sometimes you just can't avoid dealing with that. So these together here. Oh, and there's even a weird thing going on there. Okay. All right, that should seal everything together.
So,
How did I do that? That is with the merge vertex tool, which is, or it used to be say merge vertices, but it actually, I guess technically you can merge edges or faces. But I use that to just individually pull the vertices together. And then if we go back to our E selected, we should it should be back together. Um, something's looking a little wonky here. So, once in a while, I think I see what happened here. Okay, so sometimes, even the best of us get into a little bit of a jam with this stuff. And this is where the merge vertex can come in really handy. There's something that's not right with these vertices. So when I smooth it, I'm getting this weird looking thing here. It may have had something to do with the way I flipped the top one. But what I can do is drag my mouse over the vertex that I think is causing the problem and use the merge tool again. Um, and I think it's this one here too. That should smooth us out. I don't know why it's not. So there might be either a face or something. Something in there is not quite cooperating. Um, Maybe there's a duplicate face or something in there. So, once in a while you get jammed up on this stuff. If you get to that point, I mean, I could start deleting faces and clean that up. But for the purpose of what we're trying to do here, let's instead take a look at what this looks like in the cross section. And again, I'll duplicate that so that we have our whole one available to us. Okay, and it's because okay, we'll work with that. So because I uh, created that section out here, what we can do is if we want to see the connections between the cristae, we can do something more like that. And then if we want that to look like a cut edge, we can come here again. By keeping everything really simple, we can snap this to the grid. That'll flatten our edge out. And we can bring that back down. Do something like that. And other than the little gobbledygook that I got into here, oh, there we go. All right, so I'm kind of glad this is happening because we're in a spot where the geometry is not cooperating. This is an example here, hopefully, where we should be able to run a cleanup on the object. And to do that, you go under Mesh, Cleanup. And what you want to do is get rid of lamina faces and non-manifold geometry first. And in this case, because this edge is hanging out here, we could maybe say edge with 0 length too, and I hit apply. And it doesn't look like it got rid of that, which is always lovely. This is just going to hang on here and try getting rid of the faces with zero geometry. Yes.
How do we know when we're supposed to clean up?
Well, it's really when you start having problems or getting messages. So actually that one I just did there worked. I turned on edges with zero or faces with zero lengths, zero geometry, and I got rid of that. So that was reading as a face. I probably inadvertently extruded it on top and without thinking about it. But I saw something that wasn't smoothing properly. Now you can see that it's better, although this little thing here is still weird. But the general rule is when you start having problems where things don't look right, especially if you're smoothing it and expecting it to smooth, and it leaves you like a weird straight edge like I was getting. It also looks like I maybe put a hole in the back of this too. You know, it's like I clearly was being a little sloppy with this one, too, which just reiterates the fact that you have to be careful. It's helpful to keep it simple because you can fix things, but you do have to be careful as you're building things out. Um, that said, though, we have something that's reasonably decent as far as there being a um, representation of the inner wall of the mitochondria, and um, so I think we're in pretty good shape here. Let's this little pinch point is something that the way we could address this maybe is to just pick these couple vertices here and say average. Aha, there we go. So that's clearly there's something going on here. And there's a big hole in the back too. Okay, um, so how do we fix this? Well, we can use our merge vertex, and it's kind of hard to see like what we're actually looking at. But I can see I've got those two and those are connected in the right direction. So I'm going to go back to merge here, and I'm going to merge these two, and I don't know what I'm getting out of having. So I'm thinking actually I'm going to merge these together here and merge those two.
What's the shortcut for merging?
Yeah, so what I'm doing is I'm using G. G repeats the last command. So as soon as I set merge for one, and that's something when you're merging vertices together, especially if you got a bunch of them, the shortcut key helps. Once you set one, then you just pick the next group, hit G, and so on. Um, all right, I might have fumbled that one a little bit. It does that actually looks all right. So we got that all cleaned up except for the big hole in the back of it here. But if I were to take this now and select my faces and say extrude, now I've got the thickness. Maybe I'll take those in. So now I've got something that's got some thickness to it here, and I can do the same thing here. And if I put the two of those together, okay, I can group those. And now we can take our mitochondria and bend it. And we just want to make sure it bends in the right direction. You can kind of see where we're going with this, where you can play around with the. And that's how you can, so once you create one mitochondria, if you create it in a vertical axis, then you can duplicate that, you can scale it, you can put two different bend handles on it and get two different mitochondria shapes. Or, it's thinking about it that way. It's like you're creating a base shape and then you can take that and manipulate it to do other things. So, smoothie R, rough ER, mitochondria. I threw a lot at you with the MASH network. You're probably going to have questions about that. That's not a problem. Happy to answer questions with that. And we'll be doing a lot more with MASH network too, so it'll sink in after too long. Did I miss any organelles that you guys had to do? We talked about lysosomes too, just briefly, and you could fill the lysosome with a bunch of smaller spheres if you're trying to show transport. Other than Charlie's mystery on the surrounding cells of the gamete there, which it'll be fun to see if we can figure out a solution for that in Maya, but I definitely know there's a way to do it in Cinema 4D. Anybody else have anything with their cell that I didn't cover?
Why are my receptors?
Oh, yeah, the receptors. Oh, we were talking about, okay, so yeah, thanks for the reminder on that. Let's just talk about how you could go about doing that, utilizing um, the MASH network as well. I've sometimes when I'm doing this, I think about like the philosophical concept that everything is a cube. Like, like it starts to become very like very meditative. Like everything's a cube, dude.
It's like
Cube. Exactly. No joke. I mean, it's very rare that I don't start with a cube for things now. The macaroni smoothie I was probably one of the first ones that didn't start with a cube, but it just started with a ring of cubes. So, um, it's a cube, dude. Um, totally cubular. So, if you have a cell surface like this and you wanted to place um, some different structures on that surface, you could subdivide that cube into a form that gives you a bunch of different, bless you, yep, bases. And I'm just picking a few different ones here. I'm going to save this as a selection set again so I can go under create sets, quick select set, and just say receptor locator so that I can keep track of it. And again, that just creates a set that we can go back to and say select, and we can always recall that selection. And then for the purpose of the receptor, let's do this. Let's create this. Actually, this is cool because so I'm not going to start with a cube this time. I'm going to make a cylinder. And I'm also going to create a cone, and let's flip that on its axis. Okay, and let's make this one. Let's create some shaders for these so we can see them. So we'll create one shader that's yellow, one shader that's green, and so what I'm going to show you here is, in addition to selecting a single object, you can actually instance multiple objects using the mesh network too. So we're going to take both of these objects here and say mesh create network. Okay, and we only see two right now. So we want to look at our distribution. Okay, there's only 2 points, so we want more than that. Okay, and you can see that it's just kind of alternated between those. The next thing we need to do is bring our cylinder or our cube back, and we want to go to our distribution node and say we want to put this on the mesh, and that's going to allow us to drag our input over here. Oop. Okay, so now you can see we've put these on the surface, but they're on the surface via scatter, so we want to um, set that to face center, and then we also want to drag our receptor locator set over here, and that will put them exactly on the face center of what we picked. And you can see now that these, if we go to our select our set here, you can see that those are on the face. What I needed to do was count the number of faces, which looks like there's 13. So I can go back to my number of points here and just say 13. But that is basically alternating um, between the two objects. There's a node that's created called the ID node. And depending on, so basically, because we have two objects, it's rotating between those. We can set it to random. We can, I don't know, fix gives us the ability to, yeah, fix is just saying you can choose one or the other. Linear just goes one, alternates one between one and the other. So random is probably a good choice here. But that's a way that you can create a simple shape, apply it to a surface. You can apply multiple shapes to a surface, so you could have multiple receptors, and you could do it that way, or you could choose two different selection sets for the surface and distribute your um, receptors evenly that way too. So, all right, that was just a little bit of talking. So I think I think that gives you enough to roll forward with everything else that you're working on. Let me see if I can Stop the recording.